Welcome back to our revision class. I recall in the last video in this revision series, I asked you to do a genetic crossing between a homozygous dominant gene and a homozygous recessive gene. And these were the combinations I gave you. So we have two capital letter B is a pair of chromosome or genetic makeup of parent 1. And this is the homozygous recessive gene. Why recessive? Because recessive genes are represented with small letters or fonts or characters, while dominant genes in genetic crossing are represented with capital letters. Okay, so in a case where you have a combination of a big B, that's a dominant pair of allele, and a small one, that's a recessive gene, you call this a heterozygous pair of gene because it has a touch of the dominant and the recessive. So we call it a heterozygous. Heter means different in Greek, while homo means same. All right, so moving away from that, we could call this blue eyes as a trait or a character we are trying to see in the next generation. We could call this black eyes. That's those that have black pupil in the eye for the homozygous gene. So we want to see through this crossing, like I gave you as a task in your notes. So you need to follow what I'm doing step by step and compare your result. So the first thing I told you is whenever we are doing these genetic crosses, we have to separate the pair of chromosomes because that's what happens when the reproduction process is taking place after the fusion of a male and female gametes. Remember, in previous classes, the chromosome is in the helical structure where different proteins or nucleotides combine. Those proteins are simply a strand of DNAs, strand of RNAs, and all that. And I recall telling you that we have different types of proteins that combines in the helical structure. We have adenine, which we always pair with thymine. We have guanine, which we always pair with cytosine. Or it can be in a reverse form. It can be C to G, G to C, A to T, T to A. But remember, A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. So those are the different proteins or nucleotides that forms a chain on the helical chromosomal structure that gives rise to different traits and characters in offspring. So the first thing to do is you separate the pairs into individual allele like this. This is another allele. Another individual allele. I'm trying to get it across, but I don't know why it's hanging. Okay, it's free now. So you bring down the second one, then you cycle it. This is what I expected you to do in the last class. You do the same with this. Bring down the homozygous pair and separate them. Same thing for those other one. So after you've succeeded in doing this, the next thing to do is to begin the pair. So this is the rule. Now, each of these pair, now this first one here must pair with this and this from the second parent. So this is parent 1, parent 2, and we call this parent gametes. So this first one here are the parent gametes. Now they are separated into individual gametes. So the first pair here must pair or multiply this and this. Then once you're done, this other one will also cross with this and this of parent 2. So let's call this parent 1 P1. Let's call this parent 2 P2 just for identification purpose. So let's start. Remember, this pair or individual pair will cross with this. So I start. You use ruler to do this so that it will be straight. So what I have here is B and small b. It gives me a heterozygous offspring because it has a capital letter B and a small letter b, and they are both different alleles. So the big B is the allele for homozygous dominant gene, while the small b is the one for homozygous allele or traits. Okay, I don't know why this is hanging, but let me see if I can get through with this. Okay, so I want to pick the pen to give me the next line of action from here. Okay, I'm trying to get it done. Okay, so it's off now. So this one goes like this. Then you pair. Then I have B again. Remember the big letter before the small letter in that order. Then I will now go for the next individual pair here from parent 1 to parent 2. So it goes this way and this way for the first crossing. So B, this is what happens in the reproductive system of the expectant mother after conception. These are the occurrences. So this is just a pictorial way to show you how these processes occurs in real life. So this is B. So the tendency we have here is that the offspring here will all turn out blue eyes individuals. So we call this the first filial generation. We label it with the symbol F1. So they are the first filial generation. The word filial is spelled F-I-L-I-A-L. Did you catch that? All right, so in the next video, I'm going to be very quick with the next crossing I'm going to do. I'm going to give you a different pair of chromosomes to cross and see if you can come up with this. Now, if you want to further take this up, you can pick one of these and one of this from parent 1 and 2 and cross. That's what we're going to do and see the output for the second filial generation. What I say is that it's just like saying from grandfather to father, then to grandchildren, and or to children to grandchildren, going all the way from one generation to another. So take your time and write this down in your biology note as we take it up from the first filial generation. Bye for now.